Hello everybody, I am Dr. S.M. Mustafizur from Lab of Coastal Oceanography, Department of Oceanography and Hydrography, Bangabandhu Sheikh Mujibur Rahman Maritime University, Bangladesh. Today I am going to talk about fisheries diversity and environmental variables in waterways of the Eastern Sundarbans mangrove forest of Bangladesh. Here my co-authors, all are undergrad senior students, and they are working in my current projects. Here is the contents of this study. Some background of the study, some methodology, very few results, and some concluding remarks. Background of the study. Sundarbans is the largest mangrove ecosystem in the world, is continuously saving the country from frequent devastating cyclones like Shidor, Ayla, or Amphan, while providing lots of ecological benefits as well as a livelihood for over 3.5 million people relying on itself. It is a unique mangrove ecosystem of considerable ecological and economic value, lying with a total area of 10,000 kilometers square, which 62% lies in Bangladesh and the rest in India. This intertidal forested wetland provides an ideal ecological health and also supports vast stocks of fish, shrimp, and edible crab, which supply a stable food and income for the coastal communities. Here you can see the location of the Sundarban. This Sundarban is situated at the southwestern part of Bangladesh, and there are major 11 rivers and over hundreds channels and tributaries are available within this river. Its exceptional biodiversity is expressed in a wide range of both flora and fauna, with 693 species of wildlife, which includes 49 mammals, 59 reptiles, 8 amphibians, 210 white fishes, 24 shrimps, 14 crabs, and 43 mollusks. Information of vulnerability of fisheries productivity and diversity toward anthropogenic and natural threats are very limited in this area. So this project was initiated to monitor the seasonal dynamics of physico-chemical parameters and ecosystem structures and find out the major threats which can alter the optimum productivity of this region. Here the methodology, the sampling slide. This is the this sampling site is for first phase, which was done in February 2020. Samples were collected from the Poshur River in the Eastern Sundarban mangrove forest. We selected the sample site with a pre-analysis on the basis of depth and other fluvial processes as per the hydrographic chart. Here you can see the red marked point, total station 9 for first phase sampling in the Poshur River. Sample materials. We have used several instruments for oceanographic data collection, like CTD, multi-parameters, GPS, water sampler, TDS meter, thermometer, soil meter, CKDs, phytoplankton net, and zooplankton net. And also we have available lab instruments like spectrophotometers, nutrient autoanalyzer, and microscope. Here is the few results, like few data which was collected at the first sampling period, like temperature, salinity, pH, and dissolved oxygen. You can see here from the station 1 to station 8 for temperature, salinity, pH, and dissolved oxygen, all have some gradual increasing pattern. Especially for salinity, you can see here number 1 station is 7.5 ppt, whereas in station 8, 27.2 ppt is a huge difference. Also temperature have some difference, and also DO and pH. Also for transparency, TDS, and electrical conductivity, the transparency is very important. You can see the number 8 station, which is near the Bay of Bengal, is very high transparent and very clear water was found there. But number 1, number 2, 3, they're very turbid water. For 5-meter physical chemical parameters, all the temperature, salinity, pH, and they all have the increasing trend. They have some different, the same pattern as like the surface water. But here is something different for diesel oxygen. Some probably they are have some noise, and this is different from the surface water. Also for TDS and electrical conductivity for 5-meter water depth, they have some increasing trend from the station 1 to station 8. Here you can see some photograph which we collected at our sampling time. You can see the figure A, the seasonal fisherman. Actually, this area is a fishing area, but for seasonally, not for all the year round. It especially in autumn, late autumn to early summer. The figure B, the fisherman is shutting out their catch, like they are find out the major commercial species from their total catch. Here we found many jellyfish. They are shutting out and throw it away to the water. That we have found that is not a good sign for a sustainable ecosystem. Here you can see the figure C is for total catch, total different species. Here the crab, the shrimp, or the small fish, or the medium-sized fish. And the figure D is for drying area. Actually, this most of the fish are drying here, and this dried fish have a huge commercial value all the country. Here, but these figures, you can see the figure A. The fishermen are catching fish near the coast, near the coast, and figure B, different type of species. We just collected this type of species for to estimate their size, their life stage, their location. Here, figure C, all the students they are working on to collect the physical parameters of the water, and figure D, they are collecting a sediment sample for a bathing community study. Here is the concluding remarks of this study. This highly productive area is housed for hundreds of species nursery ground, and very much vulnerable because of higher fishing pressure, pollution, and natural extreme events like cyclones. This is an ongoing 3 years research project funded by University Grant Commission of Bangladesh government for analyzing the dynamics of estuarine marine mangrove ecosystem related to natural and anthropogenic threats. It is planned for at least 6 season in situ data collection yearly in the waterways of the major rivers. However, for current corona outbreak, we just had one time sampling. We cannot go for remaining in situ sampling. However, we are now analyzing the nutrient profiles, which is very much important of this water body of the water sample using auto analyzer in our lab. Here's the our lab members with me. The 4 students, they are senior 4th year student. They are also doing thesis for their undergraduate certification. That's all my presentation. Thank you all.